Hi, you guys. Good afternoon, everyone. I am pleased to introduce Torbert Rocheford, the Patterson Endowed Chair and Professor of Agronomy at Purdue University. His research areas include increasing the amount of vitamin A in corn to improve nutrition in developing nations. Today, he will present a talk titled Personalized Learning and the Future of Educational Technology. So please take a moment to silence all electronic devices. Thank you and enjoy.
Thanks. Oh, I'm on. Very good. Very good. So I was just looking at the faces. Now, there's a few students in my class, and they're here only because they're going to get some extra credit by attending, so I know why you're here. But why did you come?
To listen to your talk and to see a non-negative talk.
A non-negative talk. I'm definitely on the Don side. And why did you come? Yeah. Why did you come?
Because she's your wife.
Okay. Okay, and why did you come?
I'm married to a farmer.
You're married to a farmer. Well, that's good. And one more. Why did you come?
Me?
Yeah.
It sounded interesting.
Okay. All right. So I came because I agreed to do this 5 months ago. And I like to talk about this topic, and I'm clearly on the positive side. And we'll go for about a half an hour. You can interrupt me at any time. Raise your hand. We're going to have an interactive conversation here. And so how do you do this? Where's the techno people? How do you turn this on? If I wanted to show this, could I? Don't worry. We'll worry about it later. Okay. It's kind of the same thing. So what this represents is some of the diversity of maize. How many of you seen orange corn before? All right. How many of you have not seen orange corn before? Okay. So here's an ear orange corn. So you can never say that again. But if you're asked, like on a job interview or something like that, you can't say that. Okay. This just represents some of the diversity of maize collected from throughout the world. Like this is from Argentina or Uruguay. This is Mendoza. And we collected all this diversity when we got started in this project. And it's a pretty slide, and I usually start with that. So I've slightly changed my title because it's not just maize. It can impact other crops, and it's not just vitamin A. It can impact other micronutrients like iron and zinc, which are major micronutrient deficiencies. I'll focus on vitamin A, but the future may hold advances in automation of DNA sequencing and genotyping that will impact nutritional enhancement of grain crops throughout the world. And and thank you for telling me why you came, I'm clearly on the Dawn side of things. I think there's incredible opportunity to do this faster and better and cheaper, and we actually need to. So CIMMYT is an international research center based in Mexico, but now they have extensive activities in Africa that works on maize and wheat. And I collaborate with people at CIMMYT. That's where Borlaug was, who won the Nobel Peace Prize. He was involved in the Green Revolution and enhancing the productivity of wheat in the 60s and 70s. And, okay, now, does this have a pointer? Or do we have a real one now? It's pretty high tech. Okay. What you have, the darker yellow regions, this is where you have undernourished people. They don't have enough calories in different parts of the world. And you can see Africa and Asia are the areas with the greatest amounts of malnutrition, malnourishment. Malnourishment is not enough calories. Malnutrition is you're not getting enough vitamins and nutrients that you need to live. You could get a lot of calories, but if you don't get all your essential vitamins, you can die. So we differentiate those. So this here kind of parallels the last one, and the darker orange indicates a more intense problem of vitamin A deficiency, and the intermediate colors, it's not as bad. In the lighter colors, it's less serious in that. And so that kind of is somewhat similar If you're not eating enough food, even if it's got some vitamins in it, you may have micronutrient deficiency. So they kind of go hand in hand, but not necessarily, depending upon diet and what's there. So this is a text slide that gives you some data on vitamin A deficiency. And it starts off, you can have night blindness, and it can lead to infections. It compromises your immune system and eventually result in death. And about 250,000 to 500,000 children are more susceptible to vitamin A deficient. Children, pregnant, and nursing mothers are the most susceptible to vitamin A deficiency. And half of them die within a year of going blind. And so that's prompted the vitamin A tablets or capsules or droplets to give to people. But you have to make those every year. So Harvest Plus is about biofortifying crops, breeding vitamin A in crops instead of having to make vitamin A tablets every year. Now, the U.S. Agency for International Development in Congress loved the capsules because they reduce mortality by like 20%. Mortality. You give out these capsules and people live. And so they could see a very, very real return on their investment. But they're not accessible everywhere. And there are mothers that will walk 1 or 2 whole days with their child to get one of these capsules. But it would be better if they could just grow the crops in their backyard. So biofortification is the development of food crops that fortify themselves. Hello, Raphael. How are you? This is Raphael, who is my new grad student from Mexico. He worked at CIMMYT, and he's worked on the vitamin A project. And I think he was probably very busy working hard, and that's why he just got here, got out of class. Breeding. Plant breeding is where you select crops to make them yield more, and in this case, to make them to be more nutritious. But you have to have adoption. Now, you'll notice there's white and orange corn in this marketplace, and this was close to a site. This was in Nimba, Zambia, which is in the eastern part of Zambia. And in Africa, they eat white maize. White is good, yellow is for animals, and orange they've never seen. So there's efforts to get adoption. Efficacy means it has to have an effect on your vitamin A status. But this was a new term, and it makes a whole lot of sense to me. How many of you have had a course in biochemistry? Good. Okay. So vitamin A is a very cool model system to work on because it's a biosynthetic pathway that's very well characterized. And it's a simple event that changes it from beta-carotene, which has 2 provitamin A structure. Whoops. We go back. This is pretty high tech. This is beta-carotene. And it's cleaved by the body, and this is vitamin A. But you see this simple hydroxyl group that's added here? That does not have a provitamin A structure anymore. It's not retinol. And just very, very simple little things. And over here, alpha-carotene, the double bond is put on here in the ring structure versus there. That's not a provitamin A structure. So as a model system, it was very attractive to me when I get started because it was so precise and well defined. Flip side of that, it's a royal pain to run high performance liquid chromatography. You know, you're a plant breeder and you're doing plants and maybe you get into some DNA, then you get into these analyticals. It's a pain. So we wanted to get to the genes. We don't want to do HPLC that much. We want to get to the genes. So there's 2 sides to everything. But when you get the data and the genetics, it comes together very nicely. All carotenoids are antioxidants. And just this slight difference in terms of what vitamin A does, it will not go into certain membranes. It will not go into certain pores where it functions if it just has that hydroxyl group or a different ring structure. This is the carotenoid biosynthetic pathway. Now, this is what I meant was it was very well characterized. People have been working on this pathway for close to 100 years, and in real simple ways. When I got started, some of the genes, well, they were well characterized in E. coli and yeast, and in maize, just one had been cloned. And so I wrote my first proposal, which wasn't funded because we didn't know how to do HPLC, and they didn't believe we could do the work. And frankly, I don't blame them. So we learned how to do HPLC with another lab. When I started, one gene had been cloned, and that's the gene that's responsible for the difference between, and this is a crappy, the new crop isn't yet, the difference between white and yellow is a single gene. And maize was white. It was a mutation, a gain-of-function mutation that made it be yellow. You have carotenoids in the leaves. You have to have them. They're involved in the photosynthetic apparatus. Any idea? There's carotenoids associated with photosynthesis, and there's carotenoids in your macula. Remember how I said if deficiency is blindness? What do you think a macula and a chloroplast have in common? And you can't answer because you know chemistry. But if no one else does, I'll ask you. So what do you think? And not the students in my class that I told them the answer this morning. What do you think a chloroplast? A chloroplast, there's photosynthesis going on in the sun. There's photoexcitation. You got free radicals going all over the place. You need carotenoids to quench those and protect the photosynthetic apparatus from destroying itself. So when light comes in and you see, what's happening?
Well, you're flipping a double bond in the retinol. You have retinol and you have a molecule that can change from absorbing light.
Yeah, but you're having photoexcitation, and you can have electrons being thrown around. So you have all these antioxidants in the macula, in your retina. So they're antioxidants, and they serve different purposes. Humans cannot biosynthesize carotenoids. We have to eat them, which is cool because it gives me a reason to work on plants. Now, I've mentioned people dying of vitamin A deficiency. That doesn't happen in the U.S. We have plenty of vitamin A, and your liver will store it, and you can have a big meal that has a lot of vitamin A in it. It'll store for a while. But the U.S. population is getting older. Living longer, I should say. And our diets have changed. People eat differently than they used to. And so age-related macular degeneration is increasing. Your macula is full of zeaxanthin and lutein. These are not provitamin A structures. If you were vitamin A deficiency and you ate foods with lutein and zeaxanthin, it wouldn't take away your vitamin A deficiency. So the U.S. population might benefit from more of these type of carotenoids. And in contrast to developing countries where it's the ones that have the structure that's provitamin A. And so in the developed world, we're more worried about something like macular degeneration. And what you see is when you have macular degeneration, there's certain things you can't see very well. And there's no cure for it. If you get it, you can't fix it. So it's better to prevent it. It's not proven a diet rich in foods contain carotenoids, and it's also possible the transporters don't work so well. But I eat an ear of orange corn every day. No, I don't. Okay. A lot of data here. The world population's going up. We're having more people living in the urban areas. And we need to increase agricultural productivity faster in the next 35 to 50 years than we did in the last 35 to 50 years. We're going to add more people than we did in that time. And we had the Green Revolution during this time. You know the expression, shooting fish in the barrel? Well, some things were done that were like shooting fish in the barrel. So it's going to take some pretty sophisticated technologies to keep that rate of agricultural productivity going up. And we're building houses on farmland, and we're degrading soils. So I guess we're going to need robots and artificial intelligence. Global climate change. And this is, right now I'm on a review team for the Obama administration's USAID Feed the Future program. And these data are very, very recent from the World Bank. And there is starting to be data that temperature changes are going up, and it is reducing productivity of some of our major crops. Time will tell how that holds up, but it's not unrealistic to think that this is actually the case. So you have a decline. Same acreage, same variety, doesn't yield as well when the temperature is a little bit out of what it's adapted to. And anybody who works with house plants, you know, one part of the house versus another, it's happier, too much sun, too little water, too much water. It's the same thing with the crop. And crops can't get up and walk to the other side of the country. They sort of have to stay there. So we need research on heat and drought tolerance, not just on vitamin A, because it's got to yield. And I worked on vitamin A for a long time, and now we're also working. I'm not so much working on the heat and drought, but collaborating with people that do and thinking about grain yield. Because if you develop something that yields more and has vitamins, people will take it. If it yields a little less, if you get some women farmers and they've got children, they'll go for something that yields a little less if it's good for their children. But most people want what yields the most. So we're working on yield more. Okay, I showed you these ears. I mentioned it. And did you know that, let's just say these are random ears of corn. There's about as much DNA diversity, if we kind of got them in different parts of the world, as there is between humans and chimpanzees. Corn is a very diverse species. That's why I like working with it, because you can find variation and you can select. And we tried to train the chimpanzee to help us with our DNA isolations and extractions, but it hasn't worked. And they're pretty intelligent. So, point here is DNA. DNA assays, high-throughput sequencing, high-throughput genotyping. You just hear all the time about genetic testing, genetic screening. DNA is like a chemical. It's a molecule, but it's like a chemical molecule. So it's amenable to high-throughput, high-tech assays to determine the sequence. Hello. Oh, no, it's okay. It's okay. I'm just getting to the best part. I saved it for you. So robotics can really help here. And the sequence, you hear about the DNA sequence, human genome being sequenced, plant genomes being sequenced. We now have the capacity to do that on lots and lots of plants, and it's going to become routine, where we will have the entire genome sequence of all the plants in a breeding program. So, it lends itself. So for Africa, we're using orange for acceptance, as I mentioned. White they like, yellow is for animals. They've never seen orange, and it's working. People actually do it. This is in Maputo, Mozambique, and the person in the middle is in the picture because his shirt said Chicago, so we picked him out. You can see the white shima and the orange shima there, and on the right is my son, and the other person there is an ag economist from the University of Illinois, and we did a study if people would like orange corn, and ag economist would give you all. Any ag economists in the room? Oh, good. So I can say critical things about ag economists. No, they would give you this whole sophisticated answer. The bottom line is people will, with the right education and information, will tend to prefer the Shima, particularly mothers. The least inclined were single men. So there's cultural aspects. This is the preparation of orange maize for a feeding trial in Nimba in Zambia. And these are the children that were getting ready, and these happened to have the orange maize. Some of the other ones had white maize. They also have other foods that go with it that's more typical and also gets them to come back every day for that.
What is the food that's been prepared with the orange maize?
The orange maize, this is Shima, and this actually has some meat and green chilies. XH. Oh, it's just like polenta.
Okay.
Just
They
like
call it sadza in the Southern African region is what the polenta is.
Yeah.
And then Nemoringo is with vegetables.
Grits.
Yeah.
Grits. Yeah, American grits. So growing up, how many of you had white grits and how many of you had yellow grits?
White grits.
White grits. You know, it has no vitamin A. You like yellow grits?
I discovered yellow grits like 10, 15 years ago, and I love them.
Yeah, well, they actually have an aroma. And the orange Shima actually has more flavor, and but it's what you're used to. So with orange flesh sweet potato, Mozambique, they had a drought, a flood, and a drought, and it wiped out the sweet potato crop. So they gave away cuttings, but they were orange. And most everyone ate them, except the men didn't like the texture. And so my view is the men can just die off because the little kids are growing up, and those men, you know, they'll eat the white and they won't be as nutritious. But the kids, if they grow up with it and they taste it, they actually will prefer it because it does have a flavor. But it's all what you're used to. So these are the children who were just wonderful. Okay, breeders can select for the color orange. It's easy. People can get involved. We do it out at the research farm. And you just select for ears. This one's darker than this one, so this one wins. It's not high tech. I'll come back to it because there are some things we do to select more precisely. But it can get the breeders involved versus here, take this, you know, like a transgene from some major corporation. Pathway again. Oh, so the point back here was just because I select for orange does not necessarily mean more provitamin A carotenoids. Because here's the biosynthetic pathway, and you can go, corn is mostly lutein and zeaxanthin. They're not provitamin A structures. They don't have provitamin A. Beta-carotene and beta-krypton is what we want, but it just flies by them when you select for orange. You get a little bit more. It's like a 0.15 correlation. So you get a little bit more. So that means we needed to do some genetics to stop it here and here to get more provitamin A. And that's what we did. We had the pathway, and I have a wonderful colleague, Ed Buckler at Cornell, USDA ARS. And we had a postdoc, Carlos Harjes, who was from Bolivia originally, but German was his first tongue. And we identified this gene that affected flux in the pathway here. We wanted it to come to this side. So we identified the gene and we found a weak allele that didn't work very well. So more of it comes over to this side. And so we did that. And then we wanted to stop it here. So we get it over here. And so we're doing metabolic engineering, screening, selection, call it whatever you want. But we had a pathway to use to figure out how to do it. We don't have a pathway for grain yield. So I worked on something simple. Some people say that's smart. Okay. This is the other gene. We'll just say there were transposable elements that caused them to have less messenger RNA, less enzyme. But the fact that we have alleles of genes is at the DNA level, we've got useful information for provitamin A breeding that can be automated. I mean, the automation of DNA sequencing and genotyping is very, very high throughput. And there's companies that make money doing it, so there's got to be a good reason. So that can happen. And so I just see the future's only good. We used our knowledge to select for dark orange visually, not high tech. We have this weak allele here, and we have this weak allele that stops it there. So that's the gas pedal, this is the steering wheel, and this is a brake. And that's how we got higher levels of provitamin A. And most corn has very little provitamin A. The orange flints, say from Thailand and places, were pretty good. This is an orange flint I have here right there, derived from Thailand. The breeding program was going along and was getting some stuff around here. We made some crosses that had these weak alleles, and we did molecular marker-assisted selection. And usually when you cross a 15 by a 7 and an 8 by a 6, and you cross them together, you might end up with something like 9 or 2. in general. I mean, 9 or 10. Well, we got 22. A couple lines, and that's for breeding is pretty. It was pretty mind-boggling result. This is not what we would have expected to get something that high, but that's because we selected the specific allele and we did it in a dark orange background. So we selected those alleles. We hit the target level of 15, and we were in 3/4 tropical. It has to be adapted to there, but then CIMMYT, I'm upstream, and they take it and run with it. I'm at a university in the U.S. I work with breeders that run around Zambia and have nurseries in Mexico that environments are similar. So then they run with it. And what you can see here, and you'll just have to take my word for it because these are pretty mind-boggling results. We don't get things when we just added the gene here, this favorable hydroxylase that doesn't work too well. We got a many-fold increase. And here we got a many-fold increase. But it doesn't work in all backgrounds. And I think this is like 10 or 11. This background here, this KUI synthetic, is directly related to this corn. It's corn I put together from Thailand. And it was a program they had been selecting for orange for 20-plus years, 30 years. So it was dark orange and it had more flux into the pathway. And there's something about this that worked very well. Because you can see there's other lines that, like this one here, comparable levels to start, but didn't go up nearly as much. And we want to figure that out. We want to know, why did that work so well? Meanwhile, the breeders, they care, but they don't care. They're running with it. The color orange. For acceptance, we've talked about the color orange. So we were curious, can you map quantitative trait loci are genes with small effects? Can you map genes with small effects for the color? This is visual color. This is Kristen Chandler, was a grad student with me. She now works for Pioneer Hybrid. She had bright eyes. I never could have done this. But we had all these color categories, and I won't explain the mapping structure, but it was 10 sets of F2-derived populations. So there's only 3 or 4 categories in a population, and there were 10 of them. And that made it doable. And so the answer is, can you map color scores? Yes. And the take-home message is a lot of them went to genes in the biosynthetic pathway that we knew about. And that made sense. It made good sense. So we're happy about this. And she found 6 genes related to the pathway. This was up above was 2009. The other was 2010. Slightly differences. And from our other results and those results, okay, you can see that. So this is something that I presented a year ago in March, before the dawn of doom was even conceived. And we really have the opportunity to convert white, high-yielding, adapted hybrids that have been bred for Zambia and yield well in Zambia to orange high provitamin A with a small number of loci. Now, why would we want to convert a hybrid in Zambia versus bringing something in from someplace else?
Someplace else meaning where?
Anywhere in the world other than Zambia.
Okay.
But not the North Pole, yes.
I'm guessing because it's more well adapted.
It's more well adapted. It grows there. The breeding has been done for there, only it's white. So everything is, you've got decades of research in those breeding programs.
Natural resources come out of the African continent.
The what?
The best of natural resources usually come from the African continent. And I've studied abroad in that region.
Okay. So it's there. We want to convert it. Because if you bring in materials from other places, they may have disease resistance. Texture may not be as acceptable. That's very important. If you eat Shima, okay, grits. You all happily talk, well, some of you talk about grits. How would you like to eat grits? 3 meals a day, 365 days a year. Yeah. So they're grit connoisseurs. They really can pick up on the slightest nuances in texture and taste. So we don't want to mess with that. So that's another reason. We just had a paper accepted in genetics in the last 2 days that kind of outlines the beginning of doing this, and it's called genomic prediction, so that you sequence lines and you can predict what their carotenoid phenotype is like. HPLC is a real pain. DNA sequencing is not a real pain. It's not expensive, and it's getting cheaper, and I'd rather do that. And that's kind of the whole thing. But I said, it's a quote, the companies automate things because they have to be efficient. They're competing, and if they can automate something, they do that. So the goal really is to convert most of Sub-Saharan Africa to orange maize. But it's going to take a public-private partnership. And there are other projects for like water-efficient maize, drought-tolerant maize, maize that's adapted to African soils, where large corporations are partnering. Now, why would a large corporation want to partner with small companies and small national programs in Africa? Why would they want to do that?
Because the Western ways of doing business our ways of doing business in the developing world are drastically different from Western approaches from doing business, and there are very particular sociocultural norms and behaviors that guide whether or not you will even be able to come in there as a multinational corporation to set up shop.
Okay, so but why would you want to collaborate? And they're doing this, they're providing this for free. So why?
A multinational corporation is providing it for free?
Yeah, why do you think they're doing that? It's directly related to your point. That's a start.
Right.
So if they help,
The indigenous expertise there is indigenous expertise with small businesses in Zimbabwe. Well, more so in Angola and Mozambique than in Zimbabwe, because Zimbabwe is just politically unstable post Mugabe, and Zambia is kind of somewhat a mix of Namibia and Botswana and a little bit of Zimbabwe. But the most economically stable for business are Angola, Mozambique, and parts of South Africa. And so knowing that there is indigenous expertise that is rigorous and reliable, but they don't necessarily have the funding
Right.
in order to be able to do it all by themselves. And they do need American businesses and their infrastructure in order to make it work.
okay. So the answer, I agree with everything you said. But, well, one, to be nice. Two, what did you got to say? What? You get their foot in the door. Exactly. In a nice way. Get their foot in the door to understand how these cultures and countries work, but also if they're coming in with improved varieties and improved agriculture and the overall prosperity of the country improves, they're going to be in a better position to buy their seeds down the road. So get some good PR, but yeah, I mean, Africa's a big continent. And there's opportunity. It's going to be a while to sell a lot of seeds over there. Okay, I need to move along. We make crosses between orange and white to bring in what we want. And we have segregation. We don't want the white. And we want to know which of these yellow and oranges have more provitamin A. So we do genotyping. So as I've mentioned, the sequencing and genotyping will make things go faster. As you have more and more data, we may have artificial intelligence to help us decide which plants we want and we don't want. High performance, this is highly painful. I'm coming up with this right now, but it's certainly highly painful, and it's less, and it's costly. So I haven't thought of the L, but it's very expensive. So we want to get that out of there. And so if we do enough, and when we're sequencing all the genomes, and we do the HPLC, we may get to the point where we know exactly what genes we want to get to where we want to go. And you don't have to hardly do HPLC, except, towards the end to confirm that you've done it right. Just a quick, I didn't get into robots on this one, just a quick one in terms of we make crosses between temperate donors and CIMMYT. That's what we were doing. Now we want to go in, since we know the genes, and search in locally adapted germplasm and have less of these donors because we've found some of the genes there. Lots of people to thank. Started off as a very, very small project. Then I got some funding from USAID. Then Harvest Plus came in, and that leveraged to NSF. And Pioneer has provided some genotyping support and supported a grad student who now works for Pioneer. And there's a lot of people have helped, and it's been a wonderful collaboration. So I'm going to go real fast since it's dawn of doom. This is not my work. This is other work. And we talked about yield. We talked about robots. We're starting to fly over fields and collect multispectral data to see if we can learn something about photosynthesis and how the plants are working, how we can manage them better, how we can select them better. And it's clear that some are more green than others, just from a first run. We'll find out which ones yield more. I don't know if more green yields more, but we'll find out. And this is work of Katie Rainey and Keith in agronomy and Keith Trickauer in Ag and Biological Engineering, and Melba Crawford, who's in the College of Engineering and Agronomy, and a lot of good grad students and pilots. So you can get these scans that take information. And this is kind of, you could go and visually score, but then when they get equipment that comes along that can get a more precise measurement of whatever it is they're measuring. I got these slides yesterday. Whatever it is they're measuring, someday we'll have a robot really collecting this data, and we can spend more time designing bigger and better experiments. Bigger experiments in general are better because we've been limited. And this just shows them launching a UAV and flying it over our fields. This is the Agronomy Center for Research and Education. They collect a lot of data. And I just put the robot in there. These are some of their planes that they fly. You know, you talk about artificial intelligence, but if you've ever watched 3 or 4 grad students, postdocs working on these planes and figuring out how to get it go, boy, I think it's going to be a while before robots do that. And they argue. I mean, happily argue, but I don't know if robots are going to do that. You know, they're all working together here. I don't know about robots doing that anytime soon. So, but I like this plane. I thought that was a cool plane. That was the most Darth Vader-like plane of the pictures they sent me. But will you have a robot flying, you know, a drone like that someday? I don't know. I don't really care either. I just want to get data. So this soybean field, and this is pictures taken from above, and you can zoom in on it. And there's your soybean fields. And see, the resolution is not so good right here. You see it's good there? And they're working on lower flying planes, these little drones that have the higher resolution, because you need to be closer and slower to get it. And they'll get there. Those are plots. Okay, and then thermal imagery. Up here on the right, that's a different type of imagery. Here they're looking at the heat coming off, and it may be that some plants deal better with heat than others, and that affects their productivity. I don't know. I don't know the answer. They also use this to fly over. I think it's the same stuff they use to fly over and find people that were growing marijuana in their basements because the lights were on or something and throwing off heat. I don't know. But that's the future. So I can't even say the sky is the limit because they've got drones flying around in the sky. So the take-home message, we're just going to do what we're doing. Automation, DNA sequencing. It's only going to get faster, and it's already getting mind-boggling faster in the last 10 years. So it's only going to continue to be that way. Selection, we aren't that good at selecting too many genes all at once because you need a lot of plants and whatnot, but and figure out the best way to do it. So artificial intelligence may help. And we actually need this. We don't need it to move forward. We need it to stay less behind. Because if the population increases at the rate it is, we're falling behind. So we just need to do it. And that's my story.
I have a question, and maybe your grad student could answer it. Like you talked about.
So I'll step outside for a minute.
Like in Mexico, you talked about that they were, doing this kind of work in Mexico too. But like, have you tried to see, like, if people, you know, would accept an orange tortilla or an orange tamale, you know, I mean that brings it a little closer to home, you know, that, you know, this thing that in Africa, this thing is orange and maybe people won't like it, but I can, is the way that a tortilla is processed different than this dish in Africa that you're working with?
Well, masa has to have lime. You know, to make masa, you have to add lime to it and soak it.
And when you make grits, you're actually pounding up the corn.
But would you still have the health benefits from an orange tortilla?
I don't know what that process does. There are, you know, corn to table to mouth losses, and that would have to be considered. It's a cultural thing. The cool thing about Harvest Plus is they spend a lot of time on outreach and acceptance and getting to understand the cultures. So I don't know, and I don't really care that. I do care about Mexico, but I care where people are dying more. So in Africa, in Zambia, they were having a field day. The orange corn was there, and they were going to show the growers. And a few days before, the country manager for Harvest Plus, he brought enough ground orange cornmeal to make Shima for 100. And they were expecting 70, maybe at the most. So he comes, and he sees white Shima. And Eliab was like, he found one plate of orange Shima, and that was where the dignitaries and the politicians were sitting. And he goes and talks to the woman that made it. He says, "What happened? I brought you all this Shima. There's only one plate. I brought you all this orange corn." He says, "We heard it had vitamins. So we took it home and fed it to our children." So you don't need a statistical ag econ thing to show. And they used to have signs where they were growing the orange corn. But they don't do that. They put the sign up the day before the field day because people would come and steal the orange corn because they wanted it. So in that context, it's initially going well. Cultural, it takes a long time to change things. I mean, it just really does. You work with the kids. That's what you have to do.
It also
in schools.
Like it would be easier to get people here to eat, you know, orange tortilla chips or other orange corn products.
So actually, I'm starting a small. Yeah, they eat blue chips. I'm actually starting a small project, orange corn for the USA, and it's not motivated for the USA, but if people in the U.S., some start eating orange corn, that may be good PR for getting acceptance in other countries. And so the motivation is not, but there's actually some interest I have this orange corn. It's not GMO. It's all natural. And we've got an organic farmer growing 10 acres of it this summer. Yes, ma'am.
I wanted to know, have you looked at the possibilities of doing similar research in Brazil? Because if your intent is to have access to the wisdom, teachings, and agricultural expertise of people of Angolan and Mozambican descent, the largest population of those particular peoples are in Brazil, in Bahia, Pernambuco, and Salvador. And given I am a peacemaker, I come from a mixed family. My dad's family is German and Comanche Indian. And my mother's family are descendants of Indibitable Congo Africans out of this region. Okay, so I have to make the peace. Okay, so.
You say a piece of pie?
No, I'm saying no, and I don't. Sexual lasciviousness is a wounded topic with my mother. So what I am saying is that we are in the Midwest. The Mason-Dixon line is very real. Psychological woundedness around post-colonial realities is an open, pussy wound that is very uncomfortable to talk about. And talking about this region is like opening up that conversation. So right now, I'm looking at how do we move forward, because I have worked with professionals with the United States Agency for International Development that are alums of Carnegie Mellon. And that's how I got to the investment community with socially responsible investing. Moving forward, I think the best way to address that is start looking at where do dispersed and relocated African populations come into the new world, okay, with the wisdom that they have, And the closest place to that is Brazil.
Yeah, well, so Harvest Plus had a small project in Brazil, but at the time, most of the funding was from the Gates Foundation. And when Bill Gates came and started being very involved with the foundation, he took the view, and I think it was a good one. We weren't so happy. A lot of countries were getting a little bit, and he was like, do one place well. Get it to work, and then take that system. And so Zambia was the initial country, and now it's moving to Ethiopia and Zimbabwe. And it's also a function of how severe is the vitamin A deficient. In northern Brazil, northeastern Brazil, there is some, but it's variable. So I see what we eventually we'd like to do it everywhere, but the
that area in terms of the continent is very volatile.
Yeah.
It's very volatile and lacking its
Zambia's been a wonderful place to work. Okay, maybe another question or two. No. Is the cut meeting? Are we done? So was there a burning question, or did you say there was no burning questions? Okay. Oh, where? Yes. No. We got a little plot out at the acre. I've got one organic grower in Missouri growing 10 acres. We grow a little bit at the student farm. And I just selected this as orange as an academic thing. But then when this idea of growing it in the U.S., and I was in a meeting in Rwanda, and some things kind of came together. We're just starting. And organic farmers are interested, small farmers. The local foods in Kansas City is interested in it because it's not GMO, it's local, it's nutritious. So we're just starting. And we're going to go through the process to be able to blow it up and do it. So we're just starting the program. It'll come. Okay. I think. Okay. We're done. I think we're done.